everyone, and welcome. It's my pleasure to host today's webinar titled Process Scale Multicolumn Countercurrent Solvent Gradient Purification of Large Synthetic Molecules. I'm Keith Porter. I'm a business development manager at Bachem. I live just outside Charlotte, North Carolina. I work with our clients in the clinical peptide space. I'm happy to be on this webinar with my colleague, Ralph Eisenhuth, who is our speaker and subject matter expert. Ralph received his PhD in organic synthesis from the University of Karlsruhe with specialization in oligonucleotide and oligonucleotide synthesis. In 2008, Ralph joined Bachem as team leader in API manufacturing. Since then, he's worked in various positions, including director of Department Large-Scale Purification at Bachem in our Bubendorf headquarters. In his current position, he is the process manager, chromatography, and technology transfer. So the next several slides I have are going to give you a high-level overview of Bachem. First and foremost, Bachem is a leading innovation-driven technology or innovation-driven company specializing in the development and manufacture of peptides and oligonucleotides. Between our manufacturing sites in the US and Switzerland, we are equipped to handle the synthesis of peptides and oligonucleotides at any scale. From a business standpoint, we strive to develop strategic and long-term partnerships with our clients in the biotech and pharmaceutical space. And overall, our efforts contribute to the development and innovative medicines, helping patients around the world. Our company history, Bachem was founded by Peter Grog in 1971 in Liestal, Switzerland, which is outside of Basel. Currently, Peter Grog is honorary chairman of the board at Bachem AG. If we move ahead to 2018, due to the growing market demand for oligonucleotide therapeutics and Bachem's vast expertise in solid phase synthesis, Bachem made the strategic decision to invest into oligonucleotide manufacturing, which brings us to today. Bachem is a leading CDMO in tides with sales of over 500 million Swiss francs in 2021. This slide shows the 5 strategic foundations for which Bachem was built on. These are people and culture, innovation and technology, sustainability, customer centricity and service, and operational excellence and quality. At this point, Bachem is set to invest heavily over the next 5 to 6 years. This is to account for the expected demand in tides, as well as to remain the industry leader in tides innovation and manufacturing. So to bring us to the bulk of the presentation, I'm going to hand it over to Ralph Eisenhuth. A little bit of housekeeping. You'll see at the bottom of the screen, there's a Q&A tab. So if you simply click on this tab and post your questions at the end of this webinar, I will then ask the questions, present them to Ralph, and you'll get your answers. Ralph?
Thank you, Keith. So basically, today I will tell you about our journey to a large-scale MCSGP manufacturing. To understand how we actually ended up with the first 2 large-scale system in the industry, you have to understand how the peptide and oligonucleotide markets changed over the years, the very important green chemistry considerations, and then we will show you what we did to actually get to the systems and the additional work we had to do to be ready for large-scale MCSGP. Basically, over the last, I would say, 5 years, something significantly changed in the peptide market. We saw the advent of oral and inhaled peptide drugs. There is a general increased interest in peptides, mainly diabetes, obesity, and you see the first peptides moving into NASH. So this led to a significantly higher demand for general peptides, and we see products where we have several 100 kilograms of API per year. We actually have data for that. This is the average batch size growth from 2018 to 2021. If you take 2018 as 100%, you see that the average batch size actually more than doubled in 2 years. And this trend continues until today. This is one of the recent publications from Bachem where we informed that we had signed a large volume contract for peptides. So you can imagine the overall sales volume will be around 150 millions in 2024. So this is actually a multi-ton product, and the large volumes that everyone was talking about some years ago are actually happening right now in our facility. And the same happened and at the end of 2021, where Novartis got the FDA approval for inclisiran, a cholesterol-lowering drug. And this is the first oligonucleotide that really has a multi-ton potential, mainly because the average yearly dose of this specific compound is a little bit more than 500 milligrams. And in the USA, they have at least 1 million potential customers. And with 500 milligrams, you can do the math. You need 1 kilogram for 2,000 people and 1 ton for 2 million people, which would be a market uptake of roughly 12%. So the multi-ton oligonucleotides are really reaching the market. Of course, we anticipated that, and we are investing heavily in additional production capacity. In the upper left corner, you see the drawing of our newest manufacturing building. It will be building K. The construction started in 2021, and it will more than double the floor space for tides production at the Bubendorf facility. It will be operational at the end of 2023. In the middle here is our current large-scale manufacturing building. It has 2,900 square meters. And this is where we are doing the large-scale manufacturing of oligonucleotides and peptides. This building is getting full now, and we have some pretty impressive data regarding that. So basically what you see here is another growth chart. This time it's for column, the total column diameter for large-scale columns, meaning we added all the column diameters together. In 2017, we had 100%, and in 5 years later, we increased the overall column diameter by a factor of 2.7, which is quite impressive. And the even better thing is nearly 20% of this diameter is actually already MCSGP because we installed 2 systems in 2021. Before you start large volume productions, you have to think about the environment. And of course, something that is often forgotten, there's a certain limit of growth for manufacturing. That is usually volumes. It's not quite known in the industry besides the experts, but I will show you that this is something that really has to be talked about. These are some numbers for the manufacturing of peptides. This is the PMI, meaning kilogram of material needed to manufacture 1 kilogram of drug substance. So for this 19 amino acid peptide, we need 11,000 kilograms of starting materials, solvents, and stationary phases, and so on. For the very important groups of GLPs, the glucagon-like peptides, which are typically used in diabetes and obesity, the numbers are even higher. There we are pretty close to 16,000, and even for a very small molecule, 8 amino acids cyclic, we need something like 4,400 kilograms of material to manufacture 1 kilogram of the drug substance. The distribution according to the manufacturing stages, meaning solid phase peptide synthesis, TFA cleavage, purification, and the final stage, strongly depends on what the purity specifications are. You see, for the GLPs, we have a rather low purity specification of 96 and 98%, and there is an even distribution between the purification and the solid phase peptide synthesis in the PMI. For the generics, which generally have a pretty high purity requirement, you see that the bulk of the solvent is actually used during the purification. So you can say that the material consumption will always be a function of the actual complexity of the product and even more of the specification that you try to achieve. And of course, the yield in the chromatographic step is a key performance indicator for green chemistry consideration because it also directly influences the PMI of the solid phase peptide synthesis. For oligonucleotides, there are actually published numbers for the PMI. That's a pretty good paper published by the Green Chemistry Group of the ACS. And here we have data for 8 oligonucleotides. Here you see the PMI, which is the kilogram per kilogram. And here's the distribution for an oligonucleotide. There generally, it seems to be a rather one-to-one distribution between the synthesis and the purification. But you can multiply the PMI number with a 43% of the purification, and you will end up with a rough estimate of the PMI for the purification for the purification step. And the average seems to be around 1,892, which is quite good actually, but this is quite obvious since I guess most of these molecules are actually developed processes. So with this number, we can actually do a little mind game and think about what happens if we scale up. Usually, until the last years, oligonucleotides were mainly used in the orphan drug application niche, meaning that 10 kilograms of drug substance per year was quite a good number actually for sales. So if you multiply the 10 kilograms with a PMI of 1,892, you end up with a waste amount of 18,920 kilograms, which is acceptable since it is usually distributed over a few months of production, and you need something like 97 kilograms of sodium bromide. So that is fine. Compared to the other chemical industry companies, this is a blip on the radar, so it doesn't matter. But now comes inclisiran, which is a multi-ton project, and to actually simulate these numbers, we have to change the axis here. Now it's tons of waste and tons of sodium bromide. And for 2 million patients, meaning a 1 ton of inclisiran, you would generate something like 1,900 tons of waste, and you would need 100 tons of sodium bromide. You have to imagine, you have to accept that these are developed processes, and the 1,892 is probably a rather lower number because the real number seems to be between 5 and 10,000. So you have to multiply this by 3 or up to 5. So these are really impressive numbers. And you can imagine if the oligo field now really takes off and Bachem is a major player and we want to be a major player in this field, we will end up something like this. In 2033, you're entering Bubendorf, and to the left, you have the Monte natrium bromide, as the local people call it, and to the right, there are the salt lakes of Bubendorf. This is just a joke, but the numbers are really high. And to actually be able to manufacture such large amounts, we have to really intensify the process and reduce solvent consumption. So green chemistry always goes hand in hand with increased capacity. So how do you increase the yield of a typical purification? For peptides, it's rather easy. There we have something that is called side cut recycling, meaning that we prepare the crude solution, that we have the feed solution, then we do a purification. We take the material that is within specifications and put it into the second dimension, but we also store the material that contains product, but is not pure enough to reach the final specification. Then we repurify it in the same dimension. This is standard operation in the peptide field. We've been doing it for something like 30 years. And the impact is quite impressive. These are generics numbers that we are allowed to share. And yeah, there you see that the yield increases between 14 and 31% if you include the side cut recycling. I have to admit that for the oligo field, side cut recycling is currently not established. Also, it's quite possible. But those numbers only tell you half of those numbers are nice, but there's a price you pay for that. So basically, these are the batch purifications, the blue dots, and the orange dot is the full campaign with a side cut repurification. And the price we had to pay for that, we had to reprocess 1,600 liters of side cut fractions in 21 additional purification runs and had to make 309 additional IPCs. So side cut recycling definitely increases the yield, but it also puts a significant strain on the system, meaning we had to store those 1,800 liters somewhere in a walk-in freezer. And of course, you need an IPC team that can cope with additional IPCs. In general, for large-scale manufacturing, there's always at least one analytical chemist working together with a purification team to cope with a number of analysis that have to be performed. So MCSGP is actually quite nice because it converts the drawing to the left to something that is fully automated. What it boils down, and what I usually try to explain to people that think that it's too good to be true, MCSGP is basically automating something that we've been doing for a long time. Instead of storing the side cuts, it internally recycles the side cut fractions in the next run. We have a very nice animation for that I will want to show you now. Basically, what we have two columns that are operated in parallel. Column 1, you see 5 bands, meaning we have an early waste band, the green one. Then the magenta one is the side cut fraction in the front. The blue one is the product, and the light blue one is the side cut fraction in with the strong impurities, and then we have waste again. So while column 1 is getting eluted, column 2 is actually getting cleaned. While the product migrates down the column, the band separated, that's chromatography in the end, and the column 2 is equilibrated to actually be ready to recycle the side cut fractions. Then we discard the waste fractions in the front, and when the side cut fractions come, the early side cut fractions elute from column 1, they're diluted and stored on column 2. While the product elutes from column 1, we're actually adding new crude product to column 2, and then we recycle the strong side cuts on column 2 and go into the cleaning mode after the elution of the waste. This is something that is called a switch because in the end the column simply switched the positions, and when you do it again, you elute column 2 on column 1, then you're in the starting, and then you've done a full cycle, meaning you've gone from column 1 to column 2 back to column 1. These are important names because you will see them later on. So how did we end up with MCSGP at Bachem? Well, as always in the industry, it starts with a case study. In 2017, we actually rented a Cube 100 from Chromacon for the evaluation of MCSGP. We used tetracoxactide as a proof of principle study, since we know a lot about this purification, and we know how tedious the side cut purification for the tetracoxactide case is. These are the 1,000 liters I've shown you before. The throughput of a single shift team on one of our largest columns, the 60 centimeter columns, is actually only in the low single-digit kilogram range per week. So this is a very poor process. We have 2 orthogonal HPLC dimensions, and then we do a solid phase extraction to increase the product concentration. For the first purification dimension, we need an inverse gradient to actually achieve good separation with an acceptable column law. So what we did with the help of Chromacon, we transferred the process to MCSGP. And once we found a set point, we actually let the system run for 54 injections. I think that's something like 3 days. And we tracked the purity and the product concentration. And the nice thing about this process was that we were actually, after the first dimension, we were already nearly 1% above the target purity of 97.5. So we could fully eliminate the second purification dimension. We achieved what is usually in the brochures from Chromacon. We had the same yield as the campaign with side cuts, but higher purity. And you can imagine that if we go to lower purity, we could probably also increase the yield. The solvent consumption was already reduced by 30%. Also, we didn't do any optimization with the process, and we actually had also pretty good effects on other key performance indicators. So the total volume handled for further reprocessing, meaning that side cut fractions and the main cuts, was reduced by 90%. We increased the product concentration by a factor of 4, meaning that the throughput of the system is also significantly higher with the same freeze-dry capacity. And of course, the volume of side cuts that we had to store was 0, and this was pretty great. The data was convincing enough that we could write a report and submit it to upper management, and then we got the money to actually buy the Cube 100. And then we started further experiments. The next system was, of course, bivalirudin, something that most people know because it's one of the most shown compounds in the tides field, I would say. There the purity requirements are pretty high. It's at least 99.3% purity, so a two-dimensional purification scheme has to be applied. To achieve an acceptable yield, there's actually a really extensive side cut recycling. Nearly 20% of all runs in this specific molecule case are side cuts. We transferred the current process to MCSGP with a little bit optimization, and we had a similar result. The purity here, due to the high level, these purities are actually quite comparable because at 99.5% purity, the baseline has to be pretty good to actually have such high purity. And so these are comparable, and we had the same result at the same yield with comparable purity, but with MCSGP, and we were able to reduce the solvent consumption by something like 30%, 70%, meaning that for the MCSGP mode, we only needed 1,600 kilograms per kilogram of drug substance compared to 5,700 in the original process. Here are some numbers for the more experienced people. The throughput simply means how much crude peptide you can load, and the productivity means that how much product you get out. The usual numbers are gram per liter per hour, meaning grams per liter of stationary phase per hour. And you can with these you can convert the productivity and throughput to other systems. The more important part was actually that the throughput nearly tripled compared to a batch column on a 60 centimeter column. And we also significantly reduced the IPCs, going from 240 IPCs to something like 63. And you can even reduce it further. And we were also able to nearly double the product concentration. So these numbers were really, really good. And that was that convinced us to actually invest into the technology. So in 2020, something happened that I now in hindsight call the Big Bang. There you have the press release from YMC that a leading CMO purchased 2 systems. That was us. And this, we bought those systems. You see a little bit more of them later. And we bought a second cube for process development. So with the second cube, we started to venture into oligonucleotides because at this point, it was pretty clear that oligonucleotides will also become high volume processes. And I will now show you an oligonucleotide MCSGP process, the result of it, and later how we developed it, because that's also pretty impressive. For this specific molecule, we had developed a pretty good batch process that yielded product of a purity of nearly 94% and something like 82% yield. And the column load was okay with 459 OD per milliliter. We transferred it to the MCSGP system, and we got a pretty stable process, as you can see here, with an overlay of 30 injections, meaning 15 injections on column 1, which is usually the red race, and 15 injections on column 2. The purity was quite comparable, meaning that the process was stable. So these are the impurity profiles of Illusion 1, the blue one, and Illusion 29, the black one. We also increased the purity a little bit to 94.1 and maintained a recovery of something like 90%. And here you see really the pretty good trending of the purity. We also did long-term main cut trending to evaluate the process. I will come back to that later, and the data was also pretty convincing. Calculating the PMI was 1,300 with a pure product and 8% higher yield. This is pretty impressive. And 1,300 is actually, I would say, in the top 3 of the numbers I've shown you earlier. To the right, you see what we got out when we let the system run for something like 2 days. That's 10 grams of a synthetic oligonucleotide that was purified on 2 1-centimeter columns. So that's quite an impressive feat. With these numbers, we calculated the throughput of the system that we already installed that has 2 20-centimeter columns, and it will have the throughput of roughly 300 kilograms of drug substance per year. The system can be used with larger columns, so with 2 45-centimeter columns, which is the largest columns that you can put on the system. We will actually be able to purify more than 1.5 tons of drug substance per year. Basically, this system is inclisiran ready. And you can imagine if you take something like 60-centimeter columns, you're going into the really large-scale manufacturing of oligonucleotides. So how did we end up with this MCSGP process? I will now lead you through the development of this specific process because it's pretty nice. It's a pretty nice way to explain how you develop MCSGP. So in the beginning, you always have a batch mode. This is a UV trace of this molecule, and you define some zones that you actually want to have the system do specific things. So the light blue one is where you start the weak recycling. Then after a certain time or UV trigger, you actually start with the main cut collection. In parallel, you do the feeding. And then you stop the main cut collection and it starts a strong recycling. And then you stop the strong recycling. So these settings look pretty nice, but the more experienced people in the audience will probably know what happens if you do something like this. In this setting, everything is either collected or recycled. So there's not a lot of option for the impurities to actually go out of the system. And this is the resulting UV traces we generated. Basically, this is not something that we want. The overlay is not perfect, and you see that the peak actually gets broader and it starts to wobble, especially the blue one. The purity analysis also showed it. We had a target purity of 90% that was actually maintained, which is pretty good, but the process started to oscillate. Around injection 18, you see a drop and an increase, a drop and an increase of the impurities. This is also seen for the main cut size, which also oscillates pretty widely, and it increases, and increased main cut volume always tells you that the process is not stable. Looking at the first and 29th illusion, you see that in all cases, for the weak impurities and the strong impurities, we actually had increased levels, meaning that there was not enough chance for the impurities to leave the system in the steady state. So what we did next is first we fixed the strong impurities, meaning that we changed the trigger in the back to allow more bleed out of the system, and the resulting UV trace is shown here. In the back, we have a pretty nice overlay already for the peak, but in the front, we still have an increase in the peak. This can also be seen with the purity. The purity shows a slight decrease over time. It increased by 0.6% and the recovery was basically 100%, but the process was still not stable since there was still a slight increase in pool volume. Looking at the analytical data showed that we perfectly hit the back of the MCSGP process because this is a perfect overlay in the back impurities. And the nice thing about oligoanalytics actually is that if you have an RPIC method, the illusion profile in the analytical method is roughly comparable to the anion exchange method that we use for the purification. So the back of the peak was actually fitting perfectly, but in the front of the peak, we had still increase in weak impurities. So we fixed it. So we started to fix the front of the peak. And in this case, we simply discarded a little bit more of the front of the peak. And this is the result you saw earlier. These are 30 injections, meaning 50 injections on the left and 50 injections on the right. And the process is perfectly stable. We are now at 94.1 purity. With a recovery of 90%, this is the 8% yield that we had roughly, and the main cut trending was perfect because there's no longer a real correlation. And this is some pretty important information because it tells you how you can actually supervise the process. So when we did the oligonucleotide work, we got the first drawings from YMC, and the stuff suddenly became real. So we imagined, okay, we have this very nice system sitting in the middle of the room. In the back of the room, you see the 230-centimeter columns of the peptide system, and we want to run the system overnight. But how do you actually manage it? For that, you have to have a concept, and I will now show you in the next slide what we think will be typical supervision of the process in the future. Basically, every MCSGP process has a startup phase, meaning in the beginning of the MCSGP process, the system is not stable because it has not reached steady state. Steady state in this case means that the same quality and volume goes in and the same quality and volume goes out of the system. So you will have a startup where the purity oscillates, and at one point you will have a setting where the purity no longer changes. And once you reach this and the peak seems to be stable, you're actually in a steady state, and then you no longer have to look at each individual main cut. So once you reach the steady state, actually you can switch over to peak shape and main cut volume tracking, since this is a typical thing you would do if you would actually judge a process. So this will be the main control strategy for long-term running of the system, because it runs automatically. You simply have to check, is it still in the steady state or not? To actually get there, you have something that I personally call a long-term testing, meaning you will have a steady state after 6 injections, but you do not know if the steady state is still stable after something like 20 injections. So for this case is bivalent routine. There we did that, and there we saw something that happening after 12 hours, meaning 24 injections. Because there the peak suddenly started to oscillate, especially the purity. And after 25 cycles, a cycle is 2 switches, meaning 2 elutions, and the process stabilized again. And after another 12 to 15 hours, it started to oscillate again. This really got me thinking, and we started to investigate the process. And in the end, we could track it down to the presence of a different salt form. This is not a problem in batch chromatography, but it can be a problem in MCSGP. So we adjusted the sample preparation to get rid of the salt form, and suddenly we had a pretty robust process. As you can see here, the purity is 99.5 with nearly no trending and no correlation. And the trick to actually identifying this specific problem was injection peak. This is something that I always tell my colleagues: if a purification is not working as expected, you should really have a look at the injection peak, because you see different salt forms in the injection peak. So what we also did, we tested what happens if the system suddenly stops, because our system will stop if it thinks it runs out of solvent. So we started an MCSGP process. This is leuprolide. Here you have a purity of 99.8%. And then after we reach the steady state, in 8 cycles, we simply stopped the system after the strong recycling and the wash after it went home. And in the next morning, we restarted the system. And the resulting data is quite impressive, because the UV traces are virtually identical. And also the target purity does not fluctuate at all. This means that at least for large molecules like peptides and oligos, you actually have the option to stop the system overnight and start in the morning, or you could even exchange columns on the fly while the process is running. So now that we got that under control, we started to think about what do we do if we have a 2D process, because now we have a system that actually runs automatically, and the system will not wait for the people. So the people have actually, they need to know when to do what. So this is a typical shift plan for a 2D process. On day 1, we would, of course, start the first dimension, which is here. Then we are at the point where we actually verify the steady state. Once we got that, the team will go into the supervision mode, meaning they check if the main cut volumes are the same. While on the same day, we actually have to unload the freeze dryer and fill and start the freeze dryer again. So every MCSGP system will always be coupled with at least one freeze dryer, and they have to be synchronized. On day 2, there's not a lot to do. There, the system will still run because it's in the steady state and there's enough crude material, but we will start to run out of eluents at the end of the day. So until the shift is over, the team actually has to have prepared additional eluents for the purification. On day 3, we are doing the same because it's a pretty long process. On day 4, we would end the first dimension. meaning that the system is stopped. The main cut is homogenized, and we do a single IPC to test the purity, and then we prepare the system for the second purification dimension, which is started on day 5. On day 5, we do the same as on day 1. We start the purification, verify the steady state, and then we supervise the process. On day 6, the system is still running, and on day 7, we are done with the second dimension. We do the IPC testing, meaning analytical HPLC of the main cut, and we prepare feed solution for the day 1 of the next week. And then we prepare the switch over to the first dimension. So you see, this is something that has to be planned pretty good, or the system will stop, and we do not want this. As you saw, the development of a process is pretty straightforward. Actually, we are now at a phase where we can simply reach a steady state with 1 or 2 tries with a good batch mode. But what is actually really, what can be time consuming is the optimization of the process, because there you have to run at least 6 elutions to get into a steady state, and this is time consuming. And the traditional process characterization and especially fade and purge consumes significant amounts of material. There we have something, a cooperation with YMC regarding modeling, because modeling will actually be a very good tool for MCSGP to perform process intensification and process characterization. So here you see 4 curves. Each curve is a different load volume, meaning a column load. The lower you go, the higher the load. This load does not work because you do not reach a specification. The green line is actually the load that we're currently doing. And on the y-axis, you have the recycle time in the back of the peak. Here you see the red dot is actually where we're operating. So we are in the optimum of this load. And this is something that occurs quite often, because to actually have a high throughput, you want to have a high load. If we would operate here, the solvent consumption would be significantly higher, and the gain would not be so great. So now you saw a lot of developmental data. This is the real thing. These are 6 elutions with leuprolide. We're loading 279 grams on the 30 centimeter columns, and the time between switches, meaning between each elution, is 55%. And the productivity, meaning the amount of drug substance per kilogram per week, is something like 45 kilograms. And at least for leuprolide, this is something like, to give you a real number, this is something like 200 million doses per week. And with a 60 centimeter system, you could probably satisfy the yearly demand of leuprolide in something like 1 or 2 months worldwide. To summarize the ideal MCSGP process, also it will not be really feasible. It has no startup phase, or at least the startup phase should not generate material that is not within specifications, because you do not want to have these discussions. The steady state should be achieved as fast as possible, because you saw we will sample the first main cuts to prove that we are in the steady state. It has to be stable, especially towards disturbances in element composition, and I would say crude purity. The solvent consumption has to be low, and the product concentration has to be pretty high, because if you do not optimize for that, you're actually you're having a process that does not fit into the facility, and that's the really most important part. Or to summarize it again, if you develop a process, you do not want a Ferrari that is in the garage every 2 weeks. You want something like this, a VW Polo, something that's cheap and reliable. To summarize my talk, we are now moving into the ton range for peptides and oligonucleotides, and really MCSGP will help us to move there, because we have a pretty high degree of automation, and the footprint is pretty small for the systems. If you develop processes, really look, this is also true for batch modes. If you want to scale them up, look at the volumes of the solvents that you need, and especially the volume of the main cuts. In general, process robustness is always more important for continuous processes, especially if you move into the high volume, low margin manufacturing. Of course, all this would not have been possible without the people. So here you see to the left is our engineering team that actually built the system. And to the right, you see the process team that did the first runs, and Thomas Müller-Späth and Lars Aumann from Kummerkon, those are the guy with the masks. With that, I'm done. Thank you for listening to me. And now we can move on to the poll question.
Thanks, Ralph. So the poll question is, where do you see an innovation like MCSGP providing the most impact to your program? The options are cost savings through increased efficiency and yield, reduced solvent consumption, and scalability, the ability to achieve annual production output not previously obtained. So go ahead and enter your choice, and it should pop up for me with the results. All right, so no surprise, I think cost savings at about 70%, followed by reduced solvent consumption, which kind of ties into that, and then scalability at 15% as well. So thank you for everyone who participated in this. And we will go to our Q&A session. I saw a couple questions come in. Let me pull those up. Ralph, that was a really great talk and a lot of information. We do have a couple questions. I have a feeling most questions might be sent later after people have more time to digest all the information. The first question is, will MCSGP always be the best solution regarding purification?
No. MCSGP only makes sense if you have side cuts that are worthwhile to repurify. We have processes at Bachem where we actually have no side cuts, and there it would actually be detrimental for throughput and solvent consumption. There will never be a one-fits-all solution for oligonucleotide and peptide manufacturing, and especially purification.
Okay. One question I get, I'm actually at a conference right now at Boulder Peptide Symposium, and we were talking about scalability and scale up and the new building that's going in at Bubendorf. So one question I got face to face was, do you see a need for larger columns, and will MCSGP be a viable solution? And do you think the footprint will be smaller compared to, say, 1 meter columns or even larger versions?
That's a good one. The footprint will always be smaller because you need a smaller skid, meaning pump system. In general, the largest I imagine MCSGP system is something like 60 centimeter, and that can easily compete with a 1 meter 20 column in a full shift system. And with the MCSGP, I need something like 3 people. And in a shift system, at least in Switzerland, we are talking about something like 12 to 15 people for 1 column.
Okay. And the last question we have is, on an average 30 mer or GLP-1, what do you think are realistic increases in yield comparison to a developed batch process?
Oh, that's a good one. I would say it depends. If you do side cut recycling, then it's something like 5 to 10%. If you do no side cut recycling, it could go up to 15%. This really depends on the batch process. The good thing is for MCSGP, we have no hold time for the side cuts, which is a pretty big problem actually for GLPs most of the time. And these are the yield gains that I would expect. But I'm a little bit biased because I'm used to side cut recycling. So usually I do not see such a big gain compared to the people that do not do that.
Okay. For how many cycle lasts
a packing?
Yeah, for how many cycles does it do you get out of a typical column? Sorry, I have to reread the question. Do you need to trash them when they're older? Can you recover the column? So yeah, the packing material, I think, is what they're asking.
Usually you cannot recover RP columns. It all depends on the process. For pyrrolidine, my developmental column still worked after 200 cycles. For the oligo case, these specific columns survived at least 70 cycles, and there was no drop in yield or purity observable. But it all depends on how good the cleaning in place is developed.
Okay. If you think of the overall process from upstream downstream, how much solvent savings and solvent reduction is there for a typical oligonucleotide synthesis using MCSGP versus the solvent required for solid phase chemistry, for cleavage and all the other kind of upstream downstream processes?
This I cannot tell you, because this strongly depends on the yield and especially on the purity specification, which is always a discussion for oligonucleotides. What I can tell you is that the best batch process for oligonucleotides we made so far had a solvent consumption of 600 kilograms per kilogram.
Yeah, and I would imagine that the length of the oligo would also play a big factor to that as well. So,
yeah.
All right, we got another one. This one's kind of long. So, at the prevailing loading rates of MCSGP columns with oligo feeds, do you see a concentration shock that must be accounted for in the eluent gradient flow rate as the peak migrates through the column? And does this complicate automation? Or do you simply develop the eluent flow rate to account for this?
Oh, this is an expert question. Usually we do not see concentration shocks. Because most of the time we do anion exchange chromatography, and for AIX interactions you usually have more Gaussian peak shape compared to the Langmuir concentration shocks that you would actually observe in RP chromatography. But if it happens, we will have to adjust the system. This is not acceptable.
Okay. How do you calculate the crude loading amount for the first column and the second column?
Well, this is actually quite easy. What you do is, if you, in principle, if you want to easily develop an MCSGP process, you simply fractionate the full peak, and then you add enough material to have the same peak shape as in the last peak. This means that you calculate how much you recycle, and then you add just enough FLP to have the same load as in the previous run. This can even be done. There's a wizard in the ChromaCom Cube software that does this for this, or I personally simply use an Excel sheet. It's pretty straightforward. You just want the same loading.
Okay. So at this point, I don't have any other questions in my queue. So I think we can wrap up. So thank you everyone for your participation. I think we have a couple more slides, and we can direct everyone to Bachem's website, where you can obtain a lot more information. This webinar will be posted online. What's neat is Ralph and I did this presentation last year, so you can see the differences and how far this has come, and gives you a good idea in extrapolating this and where we'll be at in the future, as well as some of our marketing material. And this will also have instructions for how to contact us if there's any questions we didn't get to, or any questions that you think of later on. So with that, I don't have any other comments. I thank everyone for their time. Ralph, do you have any last words?
No, not really. Thank you for listening to us.
Well, thank everyone, and hope everyone have a great weekend and a good week.